Welcome to Chapter 3, Part 2. Ready to start again? Now we are going to embark upon the properties of energy, including free energy, coupled reactions, activated energy carriers, uses of activated carrier molecules, including ATP, NADH, and NADPH. So first, let's return to delta G. Delta G is known as free energy, which is essentially the amount of heat generated by the system, or in this case, by the reaction. If delta G is a positive number, the reaction is unlikely to occur because it will be creating more order in the system and will violate the second law of thermodynamics. This is when a reaction usually will need external energy, such as that provided by ATP. When a system has a negative delta G, the system is likely to proceed on its own because it will create more disorder. This is why compounds degrade over time. It's because they have a negative delta G, and as they degrade, they create more disorder. The cell harnesses these favorable reactions to drive unfavorable reactions. We call these coupled reactions. The example you are probably most familiar with is the coupled reactions of the electron transport chain and chemiosmosis. In this system, hydrogen ions are pumped across the membrane in the mitochondria, creating a gradient. These hydrogen ions, or protons, want to get back across the membrane to create more disorder. When this is allowed by the cell, they move through the ATP synthase, and this energy is used to turn the pump and thus create ATP, which would otherwise not be created due to the lack of disorder this reaction creates. Your textbook goes into more detail about the balancing of free energy. I encourage you to read through these sections to refamiliarize yourself on how the cell balances free energy. Activated energy carriers are also created through the energetically favorable reactions that occur when breaking down food. Instead of having a nicely organized hamburger, you have a mixture of amino acids, carbohydrates, nucleic acids, and lipids after digestion. Through coupled reactions, the breakdown of these energy-rich molecules will then transfer the energy to activated energy carriers, such as ATP. These carriers will then transfer the energy to unfavorable reactions, allowing for favorable reactions to occur throughout the cell. We have three main carriers you need to be familiar with: ATP, NADH, and NADPH. Your texts also discuss the role of FADH2 and acetyl-CoA, but I'm going to leave those for you to review on your own. Our main concerns are ATP, NADH, and NADPH. So how is this process actually accomplished? Let's look at the example of biosynthesis. ATP is frequently used to put two molecules together, most often in the pursuit of biosynthesis. We can see how ATP will donate one of its phosphates to a molecule to balance out the delta G of the reaction and allow the biosynthesis to occur. When ATP loses the phosphate, it becomes ADP, or adenosine diphosphate. It is still capable of giving up a second phosphate to become AMP. Remember, the goal is to increase the disorder of the environment. Besides the heat inherently lost in the transformations, the phosphate becoming free also increases the entropy of the systems. NADH and NADPH are electron carriers that are crucial for many redox reactions. They can also be considered hydride ion carriers, depending upon how you view the reactions. We will see these carriers play a large role in photosynthesis and metabolism in Chapter 13. Let's wrap up quickly with a discussion about biosynthesis. Specifically, we're going to talk about how biosynthesis needs energy, and intermediates are also utilized. Biosynthesis is the process of creating macromolecules within the body. Remember from our discussion of energy that anything that reduces the amount of entropy is not energetically favorable. This means that we must use energy to create these molecules. ATP is the most often used molecule. However, GTP, guanine triphosphate, is another molecule used to store energy. For basic reasons, the removal of one phosphate from ATP is enough to power the reaction. Remember that, along with the transformation of ATP to ADP, water is also utilized. However, for complex reactions, this is not always enough. A cell can use an intermediate pathway to get over the activation barrier. By removing two phosphates and creating AMP, or adenosine monophosphate, from ATP, the cell can release twice as much energy as it can by only releasing one phosphate. This occurs in two steps. The first is the removal of the phosphates from ATP to add them to the intermediate molecule, such as in this step right here. And this becomes the high energy intermediate, basically stating that it has a lot of energy to move through onto the next molecule. These intermediates are usually transformed pretty quickly within the cell. These phosphates are then released to create the product of the reaction or the next intermediate, depending upon the end result. Note that in the picture on the right, two AT molecules are converted to two ADP molecules instead of one ATP molecule being converted to AMP. In general, this is more favorable than the creation of AMP. This is the end of Chapter 3. Hopefully this was a good refresher on the concepts associated with energy. These concepts will be used repeatedly throughout the reactions that we discuss later in this class. Make sure you have a firm understanding of the concepts. As always, please let me know if you have any questions.